Hey guys, my name is Pras, and I'm going to be talking to you about the isolation of genes. First things first, all organisms have what's known as DNA. Inside the nucleus of their cells, DNA contains the genetic information required to make proteins that help build, regulate, and maintain your body. We can define a gene as being a length of DNA that codes for a specific protein. The phrase isolating genes refers to the identification of one of these genes and identifying which protein it codes for. The main reason as to why we isolate genes and genetically modify is the ability to engineer organisms that can synthesize useful products, which in our specific situation is a catalyst known as an enzyme. That enzyme breaks down PET, which is obviously a widely used polymer, often used for plastic bottles and food containers. There are two ways we can isolate a gene. The first method involves obtaining the mRNA, which is the molecule that conveys the genetic information from the DNA to the ribosomes, which makes the monomers for proteins. We then use the reverse transcriptase enzyme, which produces the required DNA from mRNA. The other method involves adding a type of enzyme called restriction endonucleases, which make cuts in a DNA fragment at specific points to obtain the gene. Once the gene is obtained, we make cuts in the recipient organism's DNA using the same restriction enzymes and then add the gene into the DNA, leading bonds to form. The conclusion we came up with is that we can introduce this gene into certain organisms in order to break down the PET polluting our waters. Thank you for listening.
Created using Powtoon.